Hello. If by magic you watched my poetry related video, this is immediately after, like literally like an hour after. I was like, I don't feel like it anymore. I don't feel like doing this. I don't feel like doing this. But if I don't do this, if I don't lock in, I don't know what'll happen. But it just won't be good. So we have to lock in right now. I want this done this week. I want this proposal done this week. I don't want to carry this into the next bit of time. Only thing that sucks is I could finish this proposal and not yet get the scripts from these researchers. I highly doubt they're going to send it to me this week. By chance they could, but I highly doubt that they're going to send it to me this week. Even so, I need to be prepared for the moment that I get them. And I can just mention that they're already looking for it in the proposal and then just submit it anyways because I want to get a move on. And I'll just include the paper, but we have to lock in today. It's also bothering me that I don't have this connection yet. So we just need to put everything together. I'm so tired. I need a vacation. I just need a vacation. I keep being like, why? I know that, even though I have ADHD, I would say I'm a very focused person in reality and I can get a lot done. I'm actually overall pretty organized. And sometimes I'm like, is it that I have ADHD or is it that I'm horribly burnt out from the course of events of my life? Because I'm tired and I never catch a break. Like I haven't caught a break in a long time. I'm probably gonna listen to Annie. I've been really obsessed with Annie recently. I don't know why. I was never like obsessed with Annie, but I just watched the new Annie movie recently. Not new, but from like the one from like 2015 or something. And it's so good. I'm so tired. It's 1 in the morning, but I've not been working enough. You get me? I just don't feel like I've been productive enough. I stopped to clean today. Stopping to clean, no. Stopping to clean takes time. It takes time out of your day. And then it took me like two and a half hours to make sure everything was like looking good. That's 2.5 hours behind that I am right now. You know what I mean? When this is done, bro, oh my gosh. I'm gonna throw myself a freaking party. It's gonna be so good. I don't have to have everything for my proposal, because it's not like I'm expected to finish this within the course of a year, but I don't want to work on it for a full year because I'm so tired of working on it. Do you get me? Like I don't want to do this anymore. I want it to be over. I'm so tired of not having it. Like I'm just done with this in my heart. Am I done with this externally? No. But in my heart, I'm so done with it. Okay, what we're gonna do is we're gonna assemble different concepts that have to do with what I'm talking about. We're actually just gonna pull this from my proposal. Okay, so this is the there's also pieces I have to put in like text wise. I'm not gonna focus on that too much though because that can be later when I'm writing my actual final draft. Though it could help me. Sometimes there's things that I didn't like think about. Like this probably is gonna have something in it. That's probably something to do tomorrow. Oh my gosh, tomorrow, like in Annie. Tomorrow, tomorrow. I literally am obsessed with Annie right now. Okay, look, this is what we have about binding. Now let's go to the other text. There's much more about binding. It's just that I need to put it into my actual document, which is annoying. I'm getting like oh my gosh, I'm literally full of research ideas. Every time I read something new, I get another one. Sometimes I just wish my brain would stop, you get me? But it's good too at the same time. It's just, I feel like I have to understand. We might go back to ChatGPT for this. It feels like crawling back to an ex because I haven't used ChatGPT for a minute and it pestered me and pestered me about updating my payment stuff for Plus, like for my Pro account. And I just ignored it and ignored it until it went away. So I feel like I'm crawling back to my ex right now because I have a free plan and everything. So it's kind of embarrassing, but I'm like, I need you one last time, you know? My proposal it could just be a list of different suggestions. I don't have to have it done perfectly. That's the point of having a freaking supervisor. So what I might do is I just might list like different potential things that I think could be added and why. Like almost like different formations, if that makes sense. But I don't have the mental capacity for that right now. I feel like I'm getting closer though. I feel like it's here, but just needs to be like sorted, you know. This is quite literally the last thing I need before I can finish my proposal. I just need this connection so I can explain what I'm trying to do. Because without this, I can't really explain what I'm trying to do that well. All the actual neural network stuff of like how do I translate this can be through my supervisor. I can have that conversation, but I just need to have like the baseline. If not, I don't have like a full idea, you know. Okay, this is all stuff that's going to go into my neural network probably. Right here, it's like the rules of what acetylcholine does. And if I understood that better, I'd probably understand what I'm looking for better, if we're being honest. You know what? That's probably like really, really, really important stuff. So I need to remind myself what a feed forward structure is. Oh my gosh. This is actually going to require technical skill on my part. No. Because it's probably something in this compared to how. Now I have to actually find. Let's go back to my old rat study. This is, I hate when it actually quite like challenges me. You know, this project challenges me so much. And then I get to these points where I'm like, oh, I learned a lot and like I feel really good. And then things stop being as hard, you know, where like I learned and then I get like there's a point where I was like, I didn't know how to pronounce acetylcholine. And that seemed like so hard. I was like, chemicals. I don't understand chemicals. And then I got used to it and I was like, okay, this isn't like this is like at my comfort level now. Where now it's like a new level of like, oh no, I actually have to learn something more, which is these stupid feed forward structures and plasticity rules and everything. Like, no, I don't want to learn anymore, but I have to because if I want to get this done, I have to do it. I don't even know why I'm doing this actually. You know what? We have a few different papers that we can sort of look at. This is one. You know what? The other one is the first one I actually read that mentioned that. So we're gonna go back. This is like my Shayla. This is my baby. This is my favorite paper. This was the paper that made me think I wasn't insane. This is like the best. Okay, best one. But it went into everything. My highlights are so bad. Do you see how bad my highlights are? They're not good. Because I don't own an Apple Pencil. This isn't gonna work. This sucks. I just keep having these moments where I'm like, this sucks so bad. It doesn't suck. I wouldn't say it sucks at this point, but it's just like I'm like, this could fall apart. I just feel like I keep getting that sense, but we'll see. I mean, I don't think I'm not like it's not like a sixth sense. It's just like that anxiety that like there's something very off here. Or all these little loose threads. I'm like, I don't trust that, but it'll work anyways. Okay, this is like the stress response. This is like my big point where I'm like, this could go wrong because I don't have enough information yet. So even though ACH is excitatory to hippocampal pyramidal cells, the increase in ACH release was apparently not accompanied by an increase in cell excitability or synaptic efficacy in area C1. A recent study using the same stress procedure employed here simultaneously recorded excitatory postsynaptic potentials in stratum radiatum. As reported here for the amygdala and nucleus accumbens, the only significant change in the synaptic response occurred upon release from restraint, where the response was depressed for several minutes. I want to talk about that for sure. These results suggest that any increase in cell excitability that may occur in response to the release of ACH during the stressful event is either preempted or overcome by inhibitory influences in response to the stressor. So because the chemicals are going up so much, it inhibited the excitation. In addition, they suggest that in contrast to the increase in ACH and theta activity during the stressor, these effects could be mediated via the increase in movement that occurred upon release from restraining. This is pretty interesting. This has been like a back and forth. I've read multiple different papers though. I'm going to talk about this, whether it's a movement thing. I don't think that well, it's complex, but I don't think that it is a movement thing based on other papers that I've read about acetylcholine. I mean, it could be. It's like probably a mix, but I don't know. There's a lot to say, but it's interesting that the response was depressed. I'm sorry, I'm actually really tired right now, so this is probably not like the best. So now let's go to this one. Neuronal plasticity in rat hippocampus in unpredictable and uncontrollable stress. These are kind of old, but we're gonna see if we can get updated versions of this. Okay, basically in this one, it showed that rats that could escape the shock that they were given, basically, so escapable shock, they developed significantly more long-term potentiation than the rats that couldn't escape the shock. But neither really reached the levels observed in naive controls. Okay, that really didn't give me as much as I wanted it to. I'm so tired right now. Cortical attention. Just thinking everything. I'm gonna go into some of the other papers that I read, like reread recently. So ACH, our acetylcholine king to the rescue. He was in the last one too. He says it acts as argues that it acts as a pattern separation mechanism for information arising in the CA3 recurrent collateral synapsing onto CA1. So let's go here. This pattern separation thing will most definitely be important. Tonight, I'm just going to get as much information as I can. And then tomorrow, I guess, oh my gosh, tomorrow I have a meeting. But it's not until the end of the day. It's just I have a meeting and I have my actual job. My real job has been I'm so grateful for my job. I actually like my job. But it's been randomly demanding in the midst of me doing this. And maybe it's just feeling demanding because I'm doing this, where I don't even think I'm doing that great right now, but I just want to do this. You get me? This is what I'm thinking. It's like, okay, so I'm gonna write down some of these just like loose thoughts because I think they'll probably end up in what I need them to. So this is a thing, is that we're just gonna go through a lot of different things. One, ACH actually gets triggered when there's like not like a mismatch, but when there's sort of like a surprise or novelty that requires like higher arousal. It's not actually a stress chemical per se. It's actually a novelty chemical. But it's just that stress is novelty. It's just not good novelty. Or at least a stressor is a novel thing. Stressor is novelty. What else do we know? Just because there didn't seem to be a direct correlation between like higher stress equals more ACH per se. I didn't notice that. So that rules out the idea that I have like certain ideas that are getting ruled out by that. This is what this one is saying. So specifically, cortical cholinergic inputs mediate the subject's ability to detect and select stimuli and associations for extended processing and to allocate the appropriate processing resources to these functions. My question though is how, I would say. I guess if something is more arousing, we're more likely to put our attention there. So that actually is true. So if something is more arousing, we're more likely to put our attention there. I mean, that was basically in all the arousal, like the arousal studies said that, like the arousal and binding studies said that. So, when a word is seen, a binding node is primed to form connections between the episodic context and the word's meaning. That's very human centric, though. That's a problem. When an arousing word appears in a rapidly presented sequence of words, activation of other currently primed binding nodes is delayed until binding for the high priority emotional item is complete. This is from 1999. So I need to double check what's still valid. Current theories of the role of cortical ACH include the possibility that it affects discriminatory processes, increases signal noise ratios, modulates the efficacy of cortical processing of sensory and association information, controls the reception and evaluation of stimuli for their level of significance, modifies cortical responsiveness in terms of the relevance and novelty, and confines the contents of the conscious stream. So I guess it's very responsible for cortical attention, is what the theory is. This is the thing that I keep coming back to. Is like, I guess the question would be like, what is arousing? And then the arousing thing is like what's getting encoded, right? And like there's something triggering the arousing, if that makes sense. And that thing is novelty. Less novel things are not as they're not as high of a trigger, I think, for ACH from what I read. Because when the ACH is lower, it basically helps you to like, I guess, complete patterns, what like connect new memories to old memories or something. So look at this. So hippocampal violation signals are thought to drive, okay. So when expectations are confirmed, low acetylcholine levels are maintained to promote the further generation of predictions while reducing the encoding of unsurprising and thus uninformative outcomes. So I'm going to keep putting that in because it keeps linking the idea that novelty is the important thing when it comes to ACH. So let's put that in. So when expectations are confirmed, so when the CA3 inputs match the entorhinal inputs to CA1, low acetylcholine levels are maintained to promote further generation of predictions. So sometimes actually ACH isn't like completely like, it's not always at baseline, but it's sort of in the middle. I don't know how much I'll incorporate that, but basically that's so that you can still make connections sort of, but not like, you're not like 100% in a novel state. I don't know how to quite put it. I don't know if that's even 100% accurate, but if something is, if the ACH is high, it's because there's something really novel going on. Whereas if it's a bit lower, this is the thing, is that I think that in rats, it's a little bit more simplistic because rats don't have as many connections to make, they don't have as much information, I think, to put together in terms of pattern separation and completion, I would imagine. So that's why I don't think they have this issue. So this is the things that like, this is the thing that should, I think this is how it should work, like in my thing, like my neural network. If something is novel, it should attract more attention, like attract more for like imaginary attention. Abstract, I'm going to say abstract attention. Is this too complex? We'll have to ask my supervisors in the future, whoever they are. If something is novel, it should attract more abstract attention. And then because that's what's increasing the ACH, or what's triggering the ACH to increase, I would say, is that's probably realistic, because you don't just get a, like, unless you're, I mean, when you wake up in the morning, I'm sure that the ACH goes up. But overall, if you're just hanging out, there's no reason why it should go higher unless something is like happening, like, oh, what's that sound? Or like, okay, I need to leave the house, or something like that. So whatever is arousing you should be the thing that's more increasing the ACH, right? And then as the ACH goes down, because it goes down, even in both of the studies, the ACH goes down. It should there should just be like a, I don't know, there should just be a more general or comprehensive attention, you know? Like, okay, if the ACH is up, focus. if the ACH is lower, like zoom out. That's why it's actually, I think that's why that U-shaped arousal thing happens, because if you're in that medium arousal state, you can also like you can connect it to other things and stuff. And you get, this is the thing, is that they said that people that are, I'm going to actually add this when I defend this in my proposal, but people that are highly anxious actually have a smaller, have a smaller sort of picture of the world, whereas normally people have like a bigger picture. Whereas normally people extend their memories. So that shows that if something is arousing, it's like vroom, like it's zooming in like that. And so the more arousing it is, like, you know, the picture should be like a bit smaller. And if it's not arousing at all, you're not remembering anything at all. So that's why it's like, okay, the world is in general like you're awake, you're alive, you're alert. So you're remembering something and then it's like a bit bigger versus if it's like really like, whoa, you're like, vroom, zoom, like it's in there. But yeah, and then if it's nothing, then you're just not remember. If it's not arousing at all, you don't remember anything about it, right? So, oof, okay, that's already something out of the way there, and that seems pretty cool. That's gonna be interesting to see how I how do I manage this in my computational model. Is that it? Was it that was it really wasn't as hard as I thought. Okay, that's not that bad. It just requires a bit more abstract thought. But that's kind of everything put together, isn't it? The memory binding, the arousal and the binding, the ACH. Me being like all dramatic about it, it literally took me. I feel like I've been kind of avoiding this, and it didn't take me as long as I thought. I just needed to put everything together and focus. So what this is implying, so what this should imply is that, for example, the medium arousal group, for example, if I keep my old like format, I'll do something similar. They had a moment of anxiety, which should be encoded and consolidated, most likely as like that one zoomed in memory. But my high arousal group, they're gonna have a different experience because they have something maintaining their energy for a long time. For longer, I think the inescapableness would be the novel thing too. And the stress should be impacting how they process it, should be impacting how the LTP even gets like sort of, I don't know, goes down. So they're gonna have probably a pretty different experience in general, because this is the thing about their group, is that even though the ACH wasn't as high in any like in a singular moment, it was overall high for a longer period of time. So the memories were more like zoom, more focused overall, and then also impacted by the stress. Okay, okay, this is what we're attempting. I guess I'm gonna go to sleep now because it's 2 in the morning, and this is what I was trying to do. I'm just a little bit in shock that I kind of did it. Wait, I spelled maintaining wrong. Not bad, not bad. Give myself a pat on the back. Okay, okay, we're done. I'm gonna go to sleep. Well, it's the next morning. I have bedhead, but I'm just moving notes from texts that I haven't transferred over that might have more information that I could use. It's probably one of the most time-consuming parts of this process, but also important because I make a lot of new connections that way. But I wanted to write down some of the questions that I had about like what are my missing pieces or the things that are making me feel like is this going to work, because then I could look for them as I'm rereading my texts. Okay, so this is where we stopped last night. This was a good breakthrough. This is like a paragraph right here, you know? It's like a good paragraph for my actual proposal. So like I'm just gonna do questions. Such a simple question. How does binding work? I know that sounds insane because I've read many texts now on memory binding, but I don't think that I have a good answer for this in terms of how it's gonna work in my neural network, because even though I have like an abstract sense of how it works, I need to actually look in, like I need to be able to like Frankenstein it, if that makes sense. Like in my neural networks, like I need to make it happen, which requires me actually knowing what I need. Again, these are good questions to ask my supervisor, but I want to have as much as I can before I go talk to my supervisor, because I want to be done with this project so bad, first of all. But I don't want to make them answer central questions to my project. You get me? Even also just because I feel pride in my own project, and it's like it's been my learning and my work, and I feel pride in that. And it's like I want to, if they're the central elements of what I'm doing, I want that to be like I figured it out versus I had somebody else tell me how to do it, you know? But then I want help in tweaking it. But you know, I've been working on this for how long? I started this in March. I've been working on this for like 8 months now. You know, it's my project. Like I wanna get it all the way through. I've been thinking this morning about how this is insane that I did this. I don't think I stopped to think about that enough. I just started doing this and it just became like, okay, like that's just a normal thing that you're doing. Like very quickly it became just like a normal part of my life, but it's kind of insane because I just did this like I don't know, I just felt like just started doing this and then it just happened. Like I just consistently worked on it for months. Like what? I don't know, that's just kind of a shock. Also, okay, so we have one big question today, which is how does memory binding work? And how can I translate that into my neural network? I'm currently looking back over some old texts that I read about memory binding, and we're going to talk through this part together because I think it would help me. Okay, so here we have everything from yesterday, essentially. We have everything here. So I'm reading this text. I'll scroll up in my literature review so you can see the title of the text because I don't want to scroll away from the section. It is the hippocampus, place cells, and memory. Is the spatial memory system a memory space? This was a good text. It has a lot of information in it. And I'm just looking through this again. So it's talking about, it's sort of exploring the idea of well, now we're going to have to scroll up. The hippocampus is a cognitive map and it's a bit old. It's from 1999, but what is the evidence for that? And this researcher's sort of understanding of that idea. And okay, not a great explanation, but so this is a question in this section. So is the puzzle of place cells resolved simply by characterizing hippocampal neuronal activity as a mirror of the relevant stimuli and behaviors in any behavioral paradigm? This characterization fits remarkably well both with the breadth of the data described above and with Morris and Frey's 1997 suggestion that the hippocampus automatically encodes all attended experiences. How can the hippocampus perform such a task? So if I'm understanding this correctly, basically, so they're suggesting that place cells sort of show that the hippocampus also is like sort of has a job of taking in information and sort of reconstructing it or sort of recreating it in a way that reflects what's going on outside. I know that's not the best way to put it. I don't really know how to put it, but so the development of hippocampal coding. So we presume that each hippocampal cell is capable of diverse types of coding because of the different inputs these cells receive. The distribution of cortical inputs onto the longitudinal axis of the hippocampus is very broad, and the longitudinal association pathways in the hippocampus allow recurrent connections among hippocampal neurons. At the same time, different specificities would be expected to develop among cells because the cortical inputs arrive in a graded topography such that each cell receives a different combination of plastic input weights. I'm going to put this in my section in my where'd my proposal go? In my proposal about how my neural network should operate. So, okay, we propose that when particular combinations of inputs that significantly influence a cell co-occur within the same time frame of Hebbian cooperativity, the synaptic strengths of those inputs in driving the cell are altered. Consequently, this conjunctive coding for a particular combination of stimulus and behavioral features constitutes the representation of a distinct event. It is possible that the number of repetitions required to produce event codings may be quite small, and they may occur even after a single presentation under appropriate conditions, truly reflecting learning of events within a unique episode. I'm gonna I wanna understand some of the more technical parts better. So we finally are gonna go back into ChatGPT just so I can ask it, like, can you explain this more? This is ChatGPT's best thing for me. It's such a good tool for working through an academic article. I think if you give it something really, really, like, if you give it a text, if you give it a piece of a text, it's really good at working through that and explaining it back to you in a simplified way. I think that's been my favorite way of using ChatGPT. Sometimes if you just ask it like to just like give you the information like you're doing a Google search, sometimes it's not as accurate. But if you give it a piece of text, it is pretty good at like breaking down that information for you. And that's been probably my main way of like my favorite ChatGPT like function. I think more people should use that because I think we should all become, I think it's good for everybody to learn to actually read research papers, because sometimes in the media there's so much bad sort of bad reporting of science, not in like mainstream media, but there's a lot of misinformation out there. And I think that everybody would benefit from reading the actual source of the research article more. So in that sense, I think that people should use ChatGPT more for that. If you have something that you're interested in terms of like medicine, your health, whatever, it's not, I mean, we should all learn to be more literate, scientifically literate, but I think that's been my favorite way to use ChatGPT, just in general, but that's it. So I'm gonna just copy paste here this part. Oh no. I'm just gonna ask, can you break this down for me? See, because I know this is relevant and I know this is probably gonna be really, really impactful to what I'm doing. I just don't I understand for the most part what this is talking about, but I don't understand the full details. You get me? So I was like, we're just gonna ask ChatGPT to explain it better to me. I'm excited to see how my supervisors will modify this. What are they gonna sort of think? What will they add and what will they change? That used to be something that gave me like stress, like, oh, it means I'm wrong. But I'm actually excited to see how an actual genuine expert would modify this to make it like appropriate, And after basically talking to a computer program about this for the last 8 months, I'm really excited to just talk to an actual human and get that kind of human conversation about this. Is going to be fun now that I have the knowledge to sort of interact more like just deep in a deeper way. I think it's going to be super duper fun. I'm actually really excited now. I'm excited to finish this so I can get started on like actually speaking to a supervisor, because yeah, I'm just very excited to see how they're gonna like how are they gonna shape my project. The hippocampus mixes together information from across the brain and lets its neurons influence one another repeatedly. Okay, well that makes sense. This is gonna go into my proposal now, this piece, where I will then modify it afterwards. I'm actually so excited. I want to see this published really badly. And it's going to be my very first piece of scientific writing. And I'm really excited. I want to see it come to life, like, oh, now it's real, actual published work. And it's got like life to it, I'm really hyped for that. I want to see it like done. So at the same time, different specificities would be expected to develop among cells because the cortical inputs arrived and created So graded, each hippocampal neuron receives a unique mixed input strengths, meaning the strengths can change with learning. So each neuron specializes, so represents slightly different bits or combinations of information. My question, we're probably gonna ask questions, but so place cells are only for a place. So if the information is bound, is there a specific like cell for the bound information? Does that make sense? See, these are the things I need to understand. See, each neuron ends up specializing in different bits of information or combinations of information. Another question, place cells, I guess a neuron is a type of cell. Is a place cell a neuron? Simple questions I just don't know the answer to. In my head, I was imagining as like different cells having a conversation with each other or like vibrating together or something. And then because they were doing it together, the brain registered it as one thing. But I guess that's seeming maybe like it's wrong or maybe it's right. I don't know. We propose that when particular combinations of inputs that significant, oh wait, maybe that's actually gonna be right. Wait, okay, when specific inputs to neuron happen together, the neuron strengthens the connection from those inputs. Cells that fire together wire together. Okay, well, I wasn't that far off. So if inputs arrive together often enough within a short time window, the neuron learns to treat that combination as meaningful by strengthening those synapses. Okay, so I was actually that was like pretty accurate. I keep asking a question and it kind of answers my thing, the next thing. So each neuron might come to represent fire for a unique event. I saw X while doing Y. So probably what my thing would need to do first, it would first need to bind everything together. This has been a very productive set of work sessions. So you need to bind everything together first, which would include cells firing in conjunction enough to bind an episode. So that would be like a place cell and like a something else. So I think my supervisor will come in with more specialized information. Hopefully, that's what I hope. Where they can be like, yes, in like this and this language, like I don't know, like could use like fast this or something. I don't know. That's what I'm thinking is gonna happen. I think I have a pretty good framework, but I think they'll know more specialized information and be able to like guide me with the vocab that I need for the last piece of this. And behavioral features constitutes the representation of a distinct, because okay, wait, for example, like I'm starting to learn it. I'm starting to get it into my paper, but it would take things like okay, it should be messing up the feed forward structures, or the feed forward structure should be doing this and this and that to make this happen. Or like I'm still not as comfortable with that terminology and what it means and how it works, so I don't even know how to use it as well. So I think that's going to be the first conversation I should like. I mean, that'll be the important part of having a supervisor. Okay, it is possible the number of repetitions required to produce. Okay, so I just read that. So basically, the hippocampus receives a bunch of different information from different parts of the brain. Each neuron gets a unique combination of these inputs. When the inputs co-occur in time, okay, this is really good here. So I'm going to put this in for myself, this little like explanation. They need to be occurring in time, like together at this like at the same time. So the neuron then becomes tuned to that combination, effectively representing a unique event. This process can happen quickly, possibly even after a single experience, explaining rapid episodic learning. Okay, now we're going to ask my like random questions. So a place cell isn't even a special type of cell at all. It just happens to be that those cells are tuned into place, so then they become place cells, but they're not like a special thing. They're just pyramidal neurons. Okay, so instead of place cell, it would be like I sat on the bus yesterday, so is that right? What? Okay, these paragraphs describe how each hippocampal neuron receives a unique mix of cortical inputs. Because of that, some cells may end up being tuned to spatial combinations. Others may be tuned to non-spatial combinations. The tone plus the shock, object A1 will be right, person X at location Y. So it would make event cells, essentially. Okay, we're going to make event cells. If they wire together enough, do they become one thing? Okay. When inputs co-occur repeatedly, their synapses onto a neuron strengthen together. So the neuron becomes selectively responsive to that combination of inputs. So in effect, the neuron now represents that combination as a unified pattern, a single thing in memory. So this is what's meant by okay. So basically, if you're tuned in to certain, this is okay, we're gonna write this down for me, because this is the way bindings probably work. If you're more tuned into a certain element, it's more likely to get encoded. That makes sense because your brain is probably getting this signal is probably stronger because of the arousal, right? Because you're putting that attention on something. So like if I'm looking at like my laptop, but I'm looking at the document, like the document has more attention on it, and I feel like the light of the laptop is more likely to get bound than like there's like sounds and stuff co-occurring around me. But does that make sense? So it's like you're more tuned into that signal if you have attention on it, and then that's probably making it more likely to get bound, right? So that's why certain like the binding is kind of biased towards more pertinent signals. Okay, I've been thinking recently how the first, like I was saying, the very first neuroscience book I ever read was Michael Graciano's Rethinking Consciousness. It was such a good book. I really enjoyed it. It led me down this path. I didn't think that I was going to be doing anything related to cortical attention per se, even though obviously that's still in the background because that's just the background of how our brain works. But it kind of came back to cortical attention at the end of the day, which is pretty interesting because this is essentially cortical attention as well, isn't it? Where if you have your attention on something, if it's more arousing, you're more likely to sort of like, how do I put it, tune into it, and it becomes a deeper part of who you are and how your brain works, where it's like it's biasing your brain towards certain elements that are attracting more attention. So it still is cortical attention at the end of the day. So it's kind of funny because I ended up kind of making a model of cortical attention a little bit, even though it wasn't my intention. But it came back so full circle because that's literally the book that guided me down here. And then I didn't think that was going to be cortical attention, but it kind of came back to it anyways, which is so interesting. The world is pretty funny that way. I feel like that having that information in the background of my mind was like the most important information, in this project, even though it didn't come up for a long time again, but it still like kind of came back to it. Okay, you could say before learning, neuron A responds to feature X, neuron B responds to feature Y. After repeated co-occurrence, a downstream neuron C learns to respond to both together. X and Y is now one thing. Well, I mean, this is actually, I already kind of had this in mind because that's what I was trying to do before with my little neural networks. My computational models, I was trying to build on Nengo. That I wasn't hitting the mark with it yet because I wasn't as complex as it needed to be. But this is what I was trying to do with my thing before. It just wasn't hitting the mark like technically with the actual model. But now I think it will be better because I have more information. Because I wanted it to code that event together, but I think it'll be better because now I'll have a better sense of what that information should be because it's coming from different parts of the brain that operate certain ways. So I guess the cool thing about place cells isn't even just that it shows that we are doing that for place, but that we're doing that for just across the board. Our neurons get wired. I mean, I think the play actual location part was also really cool, but I think that's kind of the special thing, right? About place cells. I feel like I've been learning a lot about place cells too as I go along and just like again, when I remember reading about play cells. I don't know if it was because I was going to UCL and I saw that. I don't know if I was doing UCL. I don't know how I came across the play cell research and I saw that it had won a Nobel Prize. And I was like, why did they win a Nobel Prize for this? I know that sounds rude, but I didn't know enough yet about neuroscience. I was like, this seems so niche. I was like, that's so small. I just thought it was really small that like, oh, okay, it's just a space, it's like a specialized cell. That's not really, because usually when you see things that win Nobel Prizes, it's stuff like it's like this is a life-saving medication sort of thing, or I didn't understand the broader impact of play cells, and now I'm understanding the broader impact of play cells a lot. As I've gone through this process, I've thoroughly understood the deep impact of play cells and why they were important. But when I first read about them, I was like, this doesn't. I mean, I was like, this is really cool. But I was like, why is this winning a Nobel Prize when it doesn't seem that far off from other? Because I was like a complete. I didn't understand anything about neuroscience. I didn't know anything. So it's like, how this doesn't seem that different from other neuroscience things that I've read about that seem so much bigger on the surface. They didn't seem like it didn't seem that special on the surface that a cell is tuned to a location. I was like, okay, what's so special about that? And see, now you see it's like it's not even just it is that's really cool and really special, but it speaks so much to how our brain, first of all, it speaks so deeply to how the hippocampus works, how our brain works, and this idea that we're so tuned in that we have what this paper was saying, that it was the idea of play cells also shows that the way that our brain is sort of encoding the world isn't just like spatial, but also cognitive. Like there's a cognitive element to how our brain is operating, if that makes sense, or how our cells are encoding. First of all, that's really cool. Second of all, I guess the hippocampus as a cognitive map thing is really important, and learning about how we navigate space is important. But it also shows that our hippocampus is basically sort of mirroring back what we have outside in a way that it can actually even sort of a singular cell can be so intimately tied to a location. Does that make sense? So that whole thing is really cool. And then it comes down to stuff like this, that it's not just locations, but things like events, relationships, whatever, that we also have. I saw one paper that was so funny. It was about the Jennifer Aniston cell. Wait, let me actually pull that up. It was basically that people across the board have a specific cell that is light up specifically when people talk about Jennifer Aniston. Like, well, this was way back in 2005, so it's kind of funny. It's hypothetical. Oh wait, well that makes sense. So in 2005 was way after the play cell. But yeah, okay, so yeah, the idea that basically there's one specific cell in our brain that only lights up when people talk about Jennifer Aniston. So that our brains are just that encoded, where there's like specific cells dedicated to really specific things. That's a really, really interesting element of the play cells research. And then also play cells then contributed to all sorts of other really important research about shockwave ripples. Also just about, I've just seen the play cells just spread out in so much of the research that I've read. Memory binding also, what else? Acetylcholine even they've talked about play cells there. Just anything sharp-wave ripple related has talked about play cells. Anything to do with like learning and the encoding and consolidating phases, a lot of times they talk about play cells. So I was like, okay, I get it now. Play cells are really important. I didn't see it at first. I was like, this is okay, like what about it? But when I started reading the research, I was like, everybody is citing this paper. Like it is that important. So yeah, that's pretty interesting, but sorry to John O'Keefe that I didn't understand. I was like, I don't get it. What's special about this right now? Honestly, I think that a lot of things that win those big prizes, I feel like you have to have a specialized understanding sometimes, because even to this day, sometimes I see people win stuff in like, I don't know, molecular biology. I don't know, or like I don't know. Sometimes it doesn't click, I think, for the average person why something would be so special and so important, you know? Where then when you but if you were a specialist or if you were to actually go into the research, like you would see what's so broad and what's so important about it. Where I think that the average person, for example, when it comes to like chemistry or biology, unless you told me like this person cured cancer, like I don't really understand half the time why something is as important as it is, even though the scientific discoveries do also have broader impacts for myself, or the kind of, some things are really obvious, like for example, like I said, like vaccines, like certain types of vaccines, or I don't know, even something like the discovery of neurons in general. We were learning in my biological psychology class. It's like, oh, that's really, really big, right? It's like such a big thing where sometimes you don't really get the full scope of what's so special about a scientific discovery without having that kind of insight, you know? So, yeah, it's pretty interesting. I mean, honestly, again, before going into science at all, if you told me something was really special and really important, I'd probably believe you, but I think I just like didn't really understand the full scope sometimes. Whereas something like a few years ago, I remember reading about the fact that they found like all of the proteins, like all of the combinations of every single protein of some kind for, I don't know if it was DNA proteins or something like that, where somebody did like an AI language model and they found all of the different proteins. It's like, okay, I definitely from the get-go see the relevance and importance of that, because I don't know enough, but I'm like, all of something seems really, really important, you know? Anyways, okay. I've talked too much about this, but I think it's been really interesting to hear about the place cells, like just deeper and deeper, um, and see how important they actually are. So basically, this is how it's gonna go. I think this is all gonna go into my actual neural network. I need those scripts. I'm really excited to get those to me. It needs to happen, and it's going to happen. Okay, we're gonna keep saying it's gonna happen because it's gonna happen. I really want those simulation scripts from the researchers that I reached out to, because I want to see how they did everything. I want to understand like even more like how they put their neural network together so that I can see what's going to be different about mine. I've become such a science nerd. It's actually crazy. I was not into, I mean, I thought it was cool. I was not into science to this degree until the last few years. And I'm like, this is so fun. But I'm really excited to get those scripts and to see like what did they do and how do I think that's the other thing it's gonna be good to talk to a supervisor about. It's talking me through how those scripts worked, and then I can sort of patchwork in the things that are important to me, you know? It's probably gonna be something like, I was kind of basing mine off of like my experiment, my neural network, off of what I actually read. But I think that it's definitely when they bind the memories, my rats. I need to cite this one. I need to actually get the notes in. But I read a memory binding paper that they found their episodes something like this, like what, where, and source. It always had some kind of what element to it, or what, when, and source, something like that. So I need that's what I'm basing this off of, and so I'll probably need a what is going to be probably like the cortex, something like that. I never spelled this right. Perirhinal cortex. Where is going to be plate cells. This is the thing, is that I'm also going to have an amygdala. So the amygdala is going to be sort of like, you know what? Maybe source is more the perirhinal cortex, and what is going to be more amygdala, you know? Like, okay, where's this information coming from? Like, why am I having this emotion? It's something like this, like, why am I having this emotion? Where am I? How do I feel about it? Like, is it, or something more like, is it safe, or is it safe? And then why is this happening? Yeah, I don't know how this is gonna go, but this is back to the same thing. I love when my ideas, I'm like, oh, this sucks, I'm gonna have to drop this, and then it kind of ping pongs back in, and it makes me feel valid. Like, okay, I wasn't far off, it's just that I needed more information. So this is basically what my thing is gonna go like. And then my supervisor hopefully will have more like insight from me, but I think that's how I'm gonna basically put together my proposal. I'm gonna close this video now. Hopefully this saved and all the audio didn't get deleted like always. And I'm gonna keep putting my text together. Okay, I have a question I'm really curious about. If someone happens to know the answer to this, you should comment. So I was reading about this more, the Jennifer Aniston cell, just on the side, and this whole grandmother cell thing. So apparently, it's the grandmother cell, sometimes called the Jennifer Aniston cell, is a hypothetical neuron that represents a complex but specific concept or object. Basically, it activates when a person sees, hears, or otherwise sensibly discriminates a specific entity such as their grandmother. So rather than basically the idea of a grandmother activating a bunch of different things in a bunch of different places, it's the idea that there's like a cell specific to that, okay? Apparently it was a joke. That was like a joke thing in the beginning, that this person, this scientist coined this in 1969 to make a joke. And then now people use it as a straw man or a foil. Like, it's not like a serious thing. And apparently by 2005, Ed Connor observed that the term had become a shorthand for invoking all the overwhelming practical arguments against a one-to-one object coding scheme. No one wants to be accused of believing in grandmother cells. Now, actually, the thing that I read about place cells is that apparently a lot of people, it was like a colleague of John O'Keefe who discovered the place cell. Apparently, people scoffed at the idea of a place cell until he discovered it, right? This is what I'm thinking, though. I'm like, it's kind of crazy because by 2005, place cells had already been discovered. And I know that people were already citing that the place cells by then because I saw it in papers. So why is it still, this is like genuinely curiosity. Why is it still crazy to think that there would be a grandmother cell if the place cell is kind of like a grandmother cell in a way? I mean, I guess because it's like, I don't know, like why is one thing like, oh, that's a crazy scientific discovery, but on the other hand, it's like a joke that would possibly happen. But actually, this is what I did read, is that in that year, so UCLA neurosurgeons published what they would come to call the Jennifer Aniston neuron, which is that after operating on patients who experience epileptic seizures, the researchers showed photos of celebrities like Jennifer Aniston. The patients who were fully conscious often had a particular neuron fire, suggesting that the brain has Aniston-specific neurons. And there's I guess what I was reading from the Wikipedia, there's back and forth on that. But why is it still kind of like a joke? Or like it's not serious? Even by 2005, in 2005, this guy was still, I mean, he wasn't saying it was bad. He was saying that it has become a shorthand for this idea that isn't really that reputable. But by 2005, they'd already discovered place cells. So doesn't that sort of indicate that that's not really that crazy of an idea? That's just my curious question as like a neuroscience beginner. What's so crazy? What's so crazy about that? If you know, you should comment. We're back in this random study area that I found that nobody's in at this time of day, at least not on reading week. I'm gatekeeping though. I look a little messy because I walked through the rain today, but anyways. So I have to lock in, like I have to start focusing. So like basically focusing really, really hard because I made a reward system for this month where basically if I do certain tasks, like I'll reward myself with the X or X thing. And my reward for finishing this, so I have 2 different rewards right now. If I finish this by the end of reading week, this proposal, I will buy myself a plane ticket to go, after I graduate, to go to Switzerland for like a week and just do a really cheap trip, like hostel. I want to go hiking in this one specific place. I've never been to Switzerland, so it's really been on my bucket list. So if I finish this proposal by this week, I get to go there. And if I'd finish it before my trip, I'm flying home for Thanksgiving. I basically have the nice, but just a little bit, just not as exciting reward of basically getting to go on a day trip to Cambridge. So, I mean, still nice, but I've got my heart set on going to Switzerland in April. So let's see if I can get this like really significantly more done. I've been really going in on this proposal, but the thing is that it doesn't actually have to be
Again, I think I'm mentally treating it like my rough draft because I was already sort of starting to write that, but it doesn't actually need to be that. So I don't need to have like every single source or all the information. It's just that I know that the thing is, the reason I'm saying this is because I have more things that I could add in terms of my documents, but that's sort of a process. And I don't know. As long as I get the answer right now, I'm good. Or get close enough. It was literally my dream to find this answer. This was basically obsessively all I thought about for like a long time until I started doing it. I was like, how am I gonna do this? Like, how do I get myself here without like I didn't have the experience yet? And I thought it was gonna be a really big push to get myself to do this when I was doing when I had gotten a comparative literature and societies degree, but it wasn't the push that I thought it was going to be. Like, it really wasn't as difficult. I was just like, how do I make that like that jump? That's so random, but it wasn't as hard as I thought. It just took a lot of work on my part. Like, I was like, how do I do this in school? Who's going to have faith in me to do this? Who's going to get me there? It's like, I had to, I had to get me there. Okay, so I'm just going to look at everything I have in this proposal so far and just see how do I move this around. So all of this information is going I'm talking about memory binding. So I guess we're going to talk first about memory binding in general. But I'm going to ask ChatGPT to confirm that I explain this right because this is memory binding, right? That's all I need to know. I mean, I think that's pretty clearly memory binding, but I just want to double check because they didn't use those words exactly. Okay, so I just wanted to make sure that I was right that is memory binding. Okay. What I think I'm going to go into after this is basically this. And this little area is about to close, which is really annoying. So I'm gonna have to move in like 5 minutes. But so this is actually all gonna go up here. And then I'm gonna talk about, so this is how memory is bound. But I'm gonna say differences in synaptic strength. Wait, that's actually such a massive clue for that. I don't know why it just clicked for me like that, but it is a difference in synaptic strength. Probably, right? I think that's gonna be right because maybe like one is more. Oh, wait, I don't know why I just clicked when I wrote this. So they're firing together, but is one part firing more? I might double confirm with ChatGPT that that's accurate. ChatGPT isn't really useful for me to like double check that my understanding is right. Like, okay, let me explain it to you and you tell me if I got that right. I'm writing, this is not my best writing yet. This is still very rough draft sentence, by the way. Have an impact. Very not sophisticated sentence, but that's essentially what I'm going to talk about. And then I'm going to talk about memory binding here. Again, I think that the big feeling that I keep getting throughout this project is just gratefulness, because on one hand, this has been like an intellectual, because on one hand, this has been an intellectual journey for me in terms of just learning. It's been just me enjoying science as well and adding something. But on the other hand, it's been such an emotional release. It's just such a massive emotional relief because I struggled with this so much. And I just feel like this hit of just like, it almost made me a little bit like, got like a little lump in my throat of just such deep gratefulness. People, I don't like, sometimes I think when you're writing a research article, I imagine this is coming from me not as a scientist now, but just as like a normal person. I don't know, just like an average person. But I don't think that people always know when you're writing something like this, just how much of an impact that's going to have on the average person. Because sometimes it just seems like maybe I imagine it just seems like that's just part of the job, or it's like another science experiment, or something just sort of interesting from like a scientific perspective, but it does have such massive impact on everybody. And like I just feel this memory binding paper that I've cited so much, it actually had such a massive impact on me to read it, and it was so special to have that information as someone who dealt with trauma. And it actually is genuinely making me emotional. Like, I don't know why I'm getting emotional about this paper, but this was such a good paper, such good science. And just everybody that has contributed to this project, even though it seems like sometimes it just
seems like very technical work, but it really does have such a massive impact. And I felt so grateful, like genuinely, like just deep, thorough gratefulness for every single person that has written a paper that I've cited, not just because it's going into my paper, but because this paper is happening, it's had such a massive impact on my mental health to be able to do this. I feel so much better about myself and etc. And yeah, I don't think you always 100% get to see that trickle down effect on a person's just emotional life and everyday life, but it really has made such a difference for me. And it's not just like, another research article. Like it's something that really had such a massive impact on the way that I'm going to navigate the world and how other people are going to navigate the world. And yeah, science is just so special. I've really, really grown such an appreciation for science and for scientists, and it's such important work even if you don't always see it. I think especially there's some papers that I've cited that you wouldn't, I mean, maybe you wouldn't expect to see it in this context, or it's just maybe a little bit, again, like a little bit more technical, like even like for example the ones that are just straight up like a long sort of explanation of the logistics of sharp wave ripples, where it's very, very technical information about sharp wave ripples, where I don't think the average person was thinking, well, that's going to help people with who have been through sexual abuse. Like it's just sort of this technical work, but it is, and it has such a big, like every little bit of science is so useful, and you just never really know how that's going to come into play. So yeah, just from an average person perspective, that's been one of the biggest things throughout this whole process, is just this deep gratefulness and just awe and just such respect that I feel towards each and every person's paper that I've read. I feel so much just genuinely so much respect, and just like you're like the coolest person on earth to me. If I cited you in this paper, like you're genuinely one of the coolest people on earth to me. And like every single paper I read, I enjoyed so much, and I just feel so much excitement and like etc. towards this paper and towards everything that I read. And it's just been, yeah, it's just been a really, like I said, this was my dream. I thought one day I really want to find this answer, and like this is really important to me. And I just feel grateful, because if it weren't for all of the different people that I cited, like this wouldn't exist. And there's so much already out there. And every time that I'm like, how do I get this next piece? I don't even know. And then I find like, let me pull the paper up again. I find a paper like this. This was from 2020. I'm like, you did it already. Like you're so smart. I just feel like every time I pull up a new paper, I'm like so hype again. That's crazy. So yeah, it's like, how easy have you made it for me to write this paper? You know what I mean? That there was already so much good work, so much important work on everything that I had here. It was just this one piece that was mine. So yeah. Okay, now I have to leave this study area. Okay, I'm currently in a random Leon where I just had the gluten-free chicken nuggets. Are my favorite. But basically, I have like 45 minutes probably left of Wi-Fi here. So what I'm gonna do is I'm just really quickly gonna sort of distribute everything out that I have into where I think it should go in my proposed computational model. And then I'm gonna end this video because I want to stay up late tonight finishing this, because it's Friday, so I can stay up late. And I don't feel like bringing this into the weekend to the degree that I would need to right now. So I'm just gonna do that and then do another video of me hopefully finishing my proposal. I think it's too ambitious to think I'm gonna fully finish it, but mostly finish it tonight. And that'll be the next video. So okay, let's go in my proposal. Once everything's in there, all I pretty much have to do is just shorten it, or just edit it, because it's currently too long. So I'm probably gonna make this into a bullet list. So for the memory binding, it needs to have, I'm gonna remind myself what some of this even means. I'm gonna go ahead and ask my question here. So I suddenly seeing it. Oh my gosh. I love when that finally happens, where like I can visualize it now. I think, if I'm right. So basically I'm asking, so each hippocampal neuron receives a unique mix of input strains, which is like, oh, it told me everything. I copy pasted its own explanation of a certain part about the combination of plastic input weights. So I'm doing a study on memory binding and how arousal can impact it, so that the more arousing element can be more likely to be bound. In that case, would it make sense that the cortex part with the most arousing piece of information would send a stronger signal. I don't know if that's actually accurate, but I'm gonna see. Oh, and then maybe if something, I'm guessing. Wait. This is because I'm also learning, so I want to see if my intuition that I'm building for this is getting more accurate. I'm gonna write this and then I'm gonna see if it's true. So the more arousing thing, I guess like the plasticity would favor it, maybe. I don't know if that's right. I'm just gonna see if that's right. So wait. Just to make sure I'm learning. So like more LTP. I don't know if that's the right way to put it, but it would be like reinforced. That's probably the two things that wait. Because I'm starting to develop this more. So that's probably two different things at work. So like I'm guessing at work that the more arousing thing might be more likely to get reinforced, and maybe the actual bound memory trace is impacted too. Question mark. Okay, so now introduce arousal. So it said that I was right, essentially, that basically that the more arousing thing would send a stronger signal, maybe. But how? The cortical region's output becomes stronger and more synchronized, which makes it more likely to drive hippocampal targets. Example, an emotionally arousing face, amygdala boosts this stuff, stronger input to the hippocampus. Modulation of hippocampal input gain. These things can change or gain or plasticity. It's more effective at inducing plasticity. Let's see if I can find a paper on that. Oh, and the amygdala. I have more papers on the amygdala to read too. I feel like I have so much more reading still to do, but we're gonna be up late tonight working on this. It's gonna be like a crash course is the next video. Arousal increases excitability and firing in sensory and associative cortices that are processing emotionally charged or salient stimuli. Okay, so we're gonna read these papers tonight. This is about emotion, but I do think it's still gonna explain things to me. Oh, some of these just look interesting. I'm just gonna download them for myself for later. Wait, this is actually so funny. Speaking of, this weekend I need to also do a crash learning of my statistics class information, but I only learned about violin graphs for the first time in my statistics class, and what? I'd never seen them before. I was like, this is insane. Okay, anyways. Okay, well, anyways, I'm gonna probably read that all tonight. But I think it's going to be something like that. I'm going to copy paste this in, not because I'm going to use this wording, but because, I mean, it basically just told me what I literally asked it if that was like accurate. And I like this explanation. So it's going to help me find some papers tonight, probably. And that's going to be my explanation for memory binding, but with more additional information. This is my question. I'm going to include some things that are questions. I might actually answer this question later because I have another paper. No, this is actually really solid now. I'm actually feeling proud of myself. I can't even believe this. I didn't know that I would be capable of actually doing this part. I was like, I'm lost. I have no idea, etc., etc. But it wasn't like, I think like once I get over the hump of being scared of something, and I give myself like my brain just a second to actually process what's happening, I'm like, okay, it's not as hard as I think, but it feels really hard in the moment. So it's going to be something like that. This is all going to be the amygdala section, because then we're going to go into that. I do need an entorhinal cortex is a yes. It has to do this pattern separation thing I read about. Oh my gosh. This is actually crazy. I didn't think I was going to do this at all. Okay, so anyways, so this reactivation thus warps patterns of hippocampal activity to mirror those evoked by the initial experience. I'm guessing I need this recurrent collaterals thing. I could probably read like 50 more texts for this, if that makes sense. Like I've gotten a lot from the ones I have, and I could probably read significantly more, and it would probably help me, and it would probably make it better. Also, this is really looking like real neuroscience. I don't know. It's kind of crazy too, because I didn't know any of this last year. This is insane. And this is so technical. It's actually crazy to look at. Like I get kind of shaken. Like, what? This is so technical, and I had no idea that it was gonna become this at all. I've kind of just been along for the ride, honestly. Like, you know what? This is insane, but I'm just gonna go with it. So I'm really worried that my Wi-Fi is gonna sort of cut out, and if that happens, it usually deletes every like all the sound on my videos for some reason. So this is what I have so far. This is looking incredibly complex, and I'm really shocked at myself, but happy. And happy because I feel like it's coming together in a way that's unique to me. And I was worried because I was gonna borrow a lot from these researchers that I am waiting on their scripts from. Like I was waiting on their scripts, but I was like kind of worried that I was doing using too much of their thing and it was going to be kind of like awkward or not enough of me. But I think there's a lot of me and my work coming through this now, where there's a lot of my own research getting sort of it's changing a lot of what they've done. I just use them as a sort of basis for certain rules that I wouldn't have the time to sort of find. Like all this stuff right here, probably it's going to be them. And I didn't do enough research to understand that stuff anyways. I did enough research probably. Well, I need to do more research, but to understand this stuff. So yeah, so this is like a really, really good setup right now. All this stuff is essentially done. So all these walkthroughs and stuff, I just need to edit them. The next steps are pretty much done. The intro, pretty much done. And the relevant section is everything's in there, but I just need to shorten it and make sure that everything is my writing rather than writing that I sort of copy pasted in. And then this intro is pretty much done, but I'm probably gonna go a little bit more into the acetylcholine, and then I need I've been really lazy to do this part, but I need to talk about all the different rat ACA studies, and I need to talk about fear extinction and sharp wave ripples. And I'm lazy, but I'll do it to make sure this is done. And yeah, this is like pretty, this is really, really looking good now. And I'll probably be able to fine tune this with my supervisor, whoever that's going to be. My supervisor is probably, I'll probably have more than one at the end of the day. But yeah, I just keep being shocked that I feel like I'm more capable than I think I am. Like I can solve some of these questions that I thought I wasn't going to be able to. And that's great. Bye.